CAST seminars spotlight innovative research in statistical sciences, both theoretical and applied, in Canada and beyond. They provide an opportunity to engage with leading experts and deepen our knowledge and understanding of cutting-edge statistics research. This seminar is a collaboration between the Department of Mathematics and Statistics at the University of Guelph and CANSI Ontario, the Ontario Regional Centre of the Canadian Statistical Sciences Institute. CANSI Ontario is dedicated to advancing research and training in statistical sciences through programs that foster interdisciplinary research and collaboration. I'm thrilled to introduce to you today's speaker, Dr. Lisa Strug, a highly accomplished researcher and leader in statistical sciences. Dr. Strug is professor in the Department of Statistical Sciences, Computer Sciences, with a cross appointment in biostatistics at the University of Toronto. She also is a senior scientist in the Genetics and Genome Biology program at the Hospital for Sick Children. She serves as the lead of the Canadian Cystic Fibrosis Gene Modifier Study, co-lead of the International CF Gene Modifier Consortium, and director of CANSI Ontario and academic director of the Data Science Institute at the University of Toronto. And so among her many achievements are the Tier 1 Canada Research Chair in Genome Data Science and the 2023 Senior Scientist Research Award from Cystic Fibrosis Canada. It is my great pleasure to welcome Dr. Strug, who will now give her talk, Genome Data Science Toward Understanding GWAS Loci. Thank you.
Thank you very much. You know, it's always an honor to be invited by graduate students, and so I really appreciate the invitation so much. And I hope we can have a long conversation about some of the work that we've been doing in the genome and in genome data science. I know the room here is filled with a mixture of individuals in statistics and data science, and really wanted to talk a little bit about my perspective on data science, or genome data science, and the work that we've been doing in the Canadian CF Gene Modifier Study. So when I think about data science, I think the data science includes the full data science life cycle, of course, the collection, manipulation, storage, visualization, and extraction of information. And that's really from increasingly larger and more heterogeneous and complex data. And so when we start to think about genome data science, right, we're just tailoring that to data that has a genetic component. But there are unique considerations for any data science about X, right? So in the situation of genome data science, there's information in the DNA sequences, and we need to decode that. There's also immense storage and computing challenges given the data set sizes. There are sharing challenges due to privacy and identity and ethical responsibilities. There's very little that's more sensitive than our genomes. And then there are specialized tools and algorithms and computational platforms that are tailored to studies of genomics data. And then, of course, there are all kinds of advancements in technologies and informatics that are generating really large biological data sets, for example, genomics, transcriptomics, proteomics. And these technologies are constantly changing and evolving. I know some of the sequencers we have at the Genome Center, the chemistry is changing almost every 6 months, right? And so we have to adapt and explore how to deal with the changing data itself as well. And then, of course, a very important area from my perspective is how do we integrate all of this data? We don't want to look at it in silos. We want to be able to let the different types of omics data or different types of public data that's out there around the genome to inform, right, and augment and enhance our interpretation of each other, as well as for understanding biological systems and precision. So today, what I want to do is I want to talk a little bit about genome data science that's happening in our lab around the cystic fibrosis and the Canadian CF Gene Modifier Study. So I don't know how many people in this room or online work in genomics, so I'll try to make sure that I keep it accessible, but if you have any questions or missing, just please jump in and ask. But we're going to tell you a little bit about the Gene Modifier Study. I'm going to tell you about how we do genomic data generation, and then all of the complexities that come along with that through the data lifecycle. data preparation, right? Data cleaning, all of those pieces. I won't belabor them because I know they're not fun, but they're very important, of course. And then genetic analysis, and then I'll dive into some statistical methodology development that was really necessary for us to be able to make progress in this study. So first, cystic fibrosis. So CF is a significant disease, right? That's a genetic disease. It's Mendelian, and it affects about 100,000 people worldwide. And what's complicated in CF is that it affects many different organs. So you can see manifestations of the disease in the lungs, in the intestine, in the pancreas. And this is caused by loss of function mutations in a gene called the cystic fibrosis transmembrane conductance regulator, or CFTR. And what CFTR is it's an ion channel, and it sits on the surface of our cells, and it transports chloride and other molecules like bicarbonate in and out of the cells to maintain a really important balance of salt and water. Now, when I started my career at SickKids, I right away started working on the genetics of cystic fibrosis. And I had some wonderful mentors and basic scientists and clinicians. And they said to me that CF is a simple Mendelian disease. That's what we think. You know, we know that it's caused by 2 mutations in the CFTR gene, and that's what you can see here on the right-hand side of the plot, these big dots that reflect the causal gene CFTR. And you don't have disease, CF disease, in the affected organs unless you have 2 mutations in that CFTR gene. But as you move down this triangle, what you can see are the different organs that are affected. And you can see that the complexity of the severity of disease in those organs increases. So for example, quite simple, probably just CFTR that contributes to what you see that's abnormal in the sweat duct. However, once you start to look at morbidity in the intestine, for example, you start to see that, or they were hypothesizing that there are many genes and potentially environmental factors that start to contribute to the differences in disease in that organ. So although CF is always thought to be a simple Mendelian disease, there's a lot of complexity that they were hypothesizing, but didn't know what the cause of that complexity was. So our challenge over the last 15 years was to try to understand what are these other genes that contribute to the complexity and the environment, how do they interact with CFTR, and what is their mechanism? So, we started the Canadian CF gene modifier study, and this is a huge Canadian effort. We have 37 sites across Canada that are enrolling patients that come to the various CF clinics to contribute to the study. And so you can just see here that all the different hospital sites where they're contributing. And we have now 3,300 participants from 9 provinces across Canada. And, one of the most important aspects of being able to make progress in data science, right, or to push disease research forward is to have really reliable, comprehensive data, right? Clinical information about the patients. And we're really fortunate that in the 1970s, they started the Canadian CF registry. So we are able to access this by applying to the Canadian CF Foundation to get access to the information on those 3,300 individuals that we recruited into our study. And they have all of their clinic data that's captured, cross-sectional, longitudinal data, lung function, whether they have bugs that are living in their lungs, what treatments they've been on, what's happening all across their body, the various comorbidities. We even know, what their latest COVID-19 test looked like. So, we're very fortunate to be able to have access to the registry. And I can't really stress enough how important something like this has been to be able to make progress. And here's sort of a snapshot of just how the Canadian CF population looks and what we're able to know just because this registry exists. For example, we know that of those individuals with CF that are living in Canada, 34% of them are children and 66% of them are adults. And that's astounding because if you looked at what those numbers were, 20 years ago, you were definitely much more spread with children, and individuals with CF were not living at this rate into adulthood. So it's a really important to be able to access this sort of snapshot and all of this deep clinical phenotypic information on this population, because then what we can do is we can go ahead and we can concentrate on collecting biospecimens and having the knowledge that we have rich clinical information that we can obtain from that's happening at the sites in the clinics. And we don't have to put any extra burden on those clinic directors to increase the amount of clinical information that's happening. And so the Canadian Gene Modifier Study has been focusing on obtaining that clinical information from the registry, which is why we have these clinic records here, but then obtaining biospecimens from each individual that's recruited into the study. So talking about bloods that are sent to our lab at SickKids, as well as on a subset of individuals, we get scrapes from their nose, and we're going to be able to do all kinds of different types of omics on these samples. And what we've been interested in is to build predictive models of disease severity and therapeutic response across the different organs. And now you know why that's important. We want to be able to use that data when we map those genes to see which ones are alternative therapeutic targets, because that could provide us with guidance on new medicines. And then we also want to know, when we map those genes, we want to understand what their impact is in individuals without CF. We all have those genes. If those genes are changed in some way, do they have an impact even if you don't have CF? So that's really what we've been focusing on. And here you see that we have enrolled it's actually 75% of the Canadian CF population. We get the data from the registry, we get the biospecimens, we send them to the lab at SickKids, and then there at the Center for Applied Genomics, we do all kinds of omic analysis. Okay, so now we're at genomic data generation. Okay, so just to back up a little bit. So here, right, we have a cell, okay? And what you can see is that we have the nucleus of the cell, and we have here the DNA. And you can see at the top SNPs, single nucleotide polymorphisms. So if we want to get to understand on an individual's genome at certain locations across their genome, do they have a T, a C, an A, or a G? We can evaluate that using genotyping arrays, and we can do that on thousands of people. Okay, and so that's why we have here circles of on locations there, and those locations would be fixed across individuals, and we would get a measure for each individual at each of these locations, what their genotype at that location is. And that's a cheap way to get a good spectrum, a good indicator of someone's genome without actually sequencing every single base. Whereas here, number 2, we could actually sequence every single base here. So what we would do is we would get the DNA, we would chop it up into fragments. We would sequence all the letters of the fragment, so not just genotype certain locations, but all of them. And then we put the puzzle back together to get somebody's full genome. So that would be sequencing. In addition, we might be interested in not getting someone's genome sequence or markers across their genome sequence, but we might be interested in understanding their level of a protein. And we're not doing proteomics. What we're doing is we're sequencing RNA, okay, as a proxy, because we know if we sequence RNA and we figure out how much RNA there is in an individual cell, that can work as a proxy for how much protein they have of certain genes. So here, what we can do is, instead of the DNA, we could sequence the RNA, right? And we would sequence it exactly as we did the DNA, chop it up into fragments and sequence the letters there, and how many reads of that RNA sequence we have is a measure of a proxy for the protein abundance. So these are the types of omic technologies that we implement in the Canadian CF Gene Modifier Study. And what's nice about having the mRNA, the proxy of the expression, and the DNA sequence or array is we can integrate that data too, okay? So we could say, okay, we have a SNP. It's a TA SNP. And then we could say we have a gene, and we know that there's this many mRNA reads for that particular gene. And we could say, okay, well, everybody with the T allele has this much expression of the gene, and everyone with the A allele has a much smaller amount, right? And we could say, okay, so then that means that this particular SNP, the one we obtained from the genotyping array or the sequence, is an expression quantitative trait locus. And we can test how different it is by computing a t-test or anything we may do like that, get a p-value and say, okay, this is a significant EQTL. This SNP that people with the disease have is associated with more expression of this gene than not. And so potentially this SNP is associated with disease through a gene expression. Okay, so we can integrate the data as well. That's my point. Okay, and just we're going to get back to this idea of EQTLs quite a bit. So if you have any questions, please jump in. But here's a sort of overview of the type of technologies we have already implemented in the Canadian CF modifier study. So lots of those genotyping arrays. So remember where we just genotype a couple SNPs across the genome. We have lots of that, thousands of individuals that we've genotyped. Then we have a lot of whole genome sequence, mostly these special new technologies of genome sequence, where we sequence very long molecules and a lot of really interesting statistical and informatics questions with that new technology, which is because it's really just come about in the last couple of years, and the prices have come down, so you can do it on large groups of people. So we have a big program where we're doing informatics and statistics around that new technology. That's not something I'm going to talk about today. But I'm going to talk about the technologies that are in blue here, where we use the RNA, the sequencing of the RNA and the genotype arrays. Okay, so a big part of any data science effort, right, is obtaining and preparing the data. So far I've talked about obtaining the data, right, and how important it is to make sure that we have large amounts of very clean and well curated clinical and genetic or omic or whatever type of data. Obtaining, preparing, cleaning that data is 95% of your time when you're implementing some sort of study in data science, right? And that's what this clock represents here. We get really excited about mapping genes, doing a genome-wide association study or doing all that follow-up study, but that's 5% of the time. 95% of the time are things like the clinical data, obtaining the clinical data, which I just showed you. It's the obtaining the genotype data and the RNA sequencing data, and making sure that it's clean and it's accurate, right? And then we need to do all kinds of things like, and I'll show you in a second, estimate genetic ancestry and estimate if there's individuals in your data set that are related, because that has implications on the statistical tests that you're going to implement later. So much time. And here's just an example of some of the quality control. I'm not going to go into it. I know it's not that exciting to hear about it, but I just want you to receive the message of just how much time and effort that takes. So here's the study that we're going to talk about today. And you can see all the different arrays, the platforms, the genotyping arrays on the left-hand side, and all the different QC steps that you need to do, whether you have duplicated variants, whether the call rate for a given SNP is less than 90%. You got to get rid of that. You have to then reduce all of the different SNPs that you're going to be looking at based on quality thresholds. And then you have to look at the sample quality control. You need to get rid of individuals that you recruited and you paid so much money to recruit into the study, and to do all the sequencing or the genotyping and make sure that nothing went wrong there. So these are all sorts of quality control. Like, for example, when you sequence them and you enroll them in the study, you have written down on your clinical data collection sheet that they're female, but your genome tells you they're male. We could have had a sample mishap. We spend so much time going through all of the sex mismatches just to make sure we have the right data and it's clean. And then once we feel like we've gone through and we have all the reliable SNP data and we have all the reliable sample data, Then we want to estimate an individual's genetic ancestry, right? Because we want to make sure that that we know what everybody's ancestry is. We want to make sure genetic ancestry. We want to make sure we know what everybody's sex is, so that we can account for all of these in the model as covariates. And so how we do that is that we would merge the genomic data that we have from our samples with public data of known ethnicity. Okay, so there's data sets out there where we know what the ethnicity is. I have some examples there, HapMap or the 1,000 genomes. And then we would calculate the principal components for this merged set. And here you can see that we're plotting principal components with known ancestry that we label in different colors, and our CF participants are in red. And you can see where they cluster here. And on the right here are the different colors, where you can see that we have known ancestry samples from external samples. So, Japanese, Italian, Chinese, European, so all kinds of different sets of individuals of known ancestry. And you can start to see across the principal components. Oh, look, my mouse works. Perfect. Okay. So you can start to see where they cluster. You can obtain the principal components. You can use them as covariates in your model. So that's one step of the data. We haven't gone into any data analysis about mapping genes, but only about understanding the sample that we're analyzing here. The other thing we need to know is there anybody in the sample that's related? If there are siblings in there or parent, child, offspring, then if we go later on and implement a linear regression model, that's a misspecified model because they're not IID in that model, right? So we need to be able to know whether we have correlated individuals in the data set. And so that's done as well from the genomic data, where we can estimate the relationships based on calculating whether the probability of whether two individuals share locations across their genome in an identical way. And so here you can start to see the plots of these probabilities, where up here in the top left corner, if they fall in here on the Z0, Z1, this probability of IBS 0,1, then they're a parent offspring. And we can start to understand whether we have these relationships in the data and account for them. And then once we've done all of that, remember, we only had genotyping arrays. I told you we only had a bunch of SNPs across the genome. We don't know what's in between those SNPs because we didn't sequence them. But it would be fabulous if we could get more data for free, right? If we could get more SNPs without actually sequencing them. And actually, you can, because SNPs, variants that are on a chromosome that are near to each other, are inherited together. So what you can do is you can do what's called genotype imputation. There are lots of software packages out there. And basically what they do is they take a reference population here. And here's our genotyped samples here. And you can see that for individuals that have an A, a G, and a C in those locations, most of the time, they have this TCT before that. You can fill it in. And so we have clean data. We know who's related. We know what their genetic background is. We got a reference sample that matches, that contains some of the common genetic backgrounds, and we can fill this data in. So now we just don't have a few markers across the genome. We could have 9, 10, 20 million markers across the genome now. And we can make that genotype imputation process even better. This is a study that we did a few years ago, where we said, okay, well, that's great to be able to do genotype imputation from some reference population of healthy individuals. But what if we threw some sequence in of individuals with CF, right? Would that improve the imputation? We could construct a hybrid reference of those haplotypes. And here you go. This is just a study of how you can see the hybrid versus a regular imputation reference panel, how the hybrid on chromosome 7, where CFTR, the causal gene is, does better than of course, the reference population. And actually, the hybrid does better across all the chromosomes on the genome than the reference. So we did a little data science project not on the analysis part, but on the data cleaning and the data preparation part. So there's data science questions at all levels of this sort of data science life cycle. Okay, all of that, not that exciting, right? I mean, it's cleaning, preparing a data set to map genes, which we think is really exciting, right? And that's what we get to. So what are the statistical questions? What are the complexities when we want to do a genome-wide association study? Does everybody know what a genome-wide association study is? Okay, a genome-wide association study takes all those markers that we just imputed, all those SNPs that we just imputed across the genome, and it says, okay, if I'm interested in which SNPs are associated with my lung function, which is something that I'm going to show you here, I could run a linear model. Let's say they're all unrelated individuals in the study. I could run a linear model and say, okay, is this T allele associated with how high my lung function is? And I could do a bunch of simple regressions, and I can get a p-value from those. And when I get a p-value that's really small, that tells me that that SNP is associated with lung function. And I can do that for those millions of SNPs we just imputed. And that's going to tell me the locations, the genomic locations that may be contributing to my phenotype. So we put together, as part of the International CF Gene Modifier Consortium, all kinds of different studies, okay? We have a study from France, and they collected CF individuals who are just unrelated to others in the study. Then we have a study from UNC, and we put them together with this study where these are individuals also independent, but they were sort of ascertained by having like an extreme lung function phenotype, different studies design. Then we have our Canadian CF gene modifier study. That's population-based. We didn't do anything complex. And then we have a study from Johns Hopkins University, and they collected twins and siblings. Okay, so you can imagine the complexity. How are you going to analyze all of these data sets together to try to do what I just said, which is to find SNPs in the genome that are associated with lung disease, but we want this big sample size and throw them all together. So these are some of the questions that come up, when you are trying to put these large data sets together with lots of different genomic platforms. And so in the end, we had 13 different platforms that we genotyped the samples on from all kinds of different centers. And we were analyzing almost 17 million SNPs, okay? And we ended up doing a meta-analysis, and we did a fixed meta-analysis and a random effect meta-analysis. And we minimized across the genome. So that was the model we ended up using, and we adjusted for the principal components, the genetic ancestry, we adjusted for the sex, and we knew who was related in the sample. So we could run a model, a linear model here, fixed, and a random effect meta-analysis. And finally, for each one of the models were a little bit different, of course, then we meta-analyzed them. Yes?
Visualization is like very simple, like interpreting model. So does it able to capture every?
The linear model? Yeah. Like, so are you asking, what about, why don't we do something like that's a non-linear model or all kinds of? Yeah. So, I mean, so for the different data sets, we use different types of models. We used a linear mixed model for this one because they're related, but they were all linear models that we used. We didn't use any non-linear models. We're analyzing in this particular data set about 6,000 individuals across 17 million SNPs. So we weren't doing any non-linear modeling at that time, although we did look at things like interactions and that sort of thing. And this is what we got. Okay, so what you can see here is on the X chromosome, on the x-axis, you have all the different chromosomes. They're in the order, the location on the genome. Here on the y-axis is the p-value from the association test. Remember, I said you could do a regression model with one SNP and the lung function, and it gives you a p-value on the minus log base 10 scale. It's called a Manhattan plot because you're looking for skyscrapers here, right? And so what you can see here is that many locations came up. Many locations in the genome came up, suggesting that we have 5 locations that contribute to lung function in CF. So that's very exciting. But we have no idea how a given location contributes to CF lung disease. So here are two locations here. one here on chromosome 5 and one here on chromosome X. All we know is there are SNPs over here. We don't know what their gene is, what the responsible gene is, what the responsible mechanism is. And you can zoom into these locations, and what you can start to see, here's the chromosome 5 location and the chromosome X location, each dotted one of those SNPs that I showed you before. And you can see that these start to get very complicated in there. And there's all kinds of genes under here. We have no idea which gene is responsible. And here you have the X chromosome as well. A very strange looking bunch of SNPs that are contributing in these weird patterns. The colors represent, you have a purple SNP and the colors red and orange show that they're highly correlated with each other, whereas a blue SNP means that it's not correlated at all with the top SNP over here. So these look like independent signals. Could two locations in this one region be contributing differently? We have no idea, okay? And that's where we're left after a genome-wide association study. What we know is that none of these SNPs actually have a known function that changes the protein coding sequence of the gene. So they don't change, they don't have an obvious function, okay? But we do know that all of the genes, all but one of the genes are expressed in the lung, and that we do know that one gene in this location, this one, SLC9A3, has been a known candidate gene. They've done studies. It is also a transporter. And I remember I told you CFTR is also a transporter. So you can start to look at these genes and think, oh, well, that makes sense, or maybe, but that's about all you have. So the question becomes, how does this chromosome 5 locus impact CF lung disease? And this is where the statistical methodology piece comes into play. We don't know whether the SNPs that are contributing to the CF lung disease are doing so through potentially gene expression, because SNPs that impact gene expression, are not usually sitting in they're usually sitting in regulatory areas of the genome, not in the protein coding regions of the genome. So the first thing we need to do is we need to say, okay, if that's our hypothesis that SNPs that are contributing to gene expression impact CF lung disease, well, these SNPs that are associated with CF lung disease, they better also associate with gene expression. So let's make a visualization tool. Okay, so that's what we did. What we have here, these SNPs here in this zoom in plot on the chromosome 5 locus here, these are the SNPs that were in the Manhattan plot that show that they were associated with lung disease. And this gray line here takes the same SNPs in the region, the same dots, and it asks, what is the evidence that these SNPs in this region are EQTLs for the SLC9A3 gene? And we can just draw a smooth line between the p-values for association of the SNP with the gene expression, okay? And so we call these co-localization plots. We're asking the question, the SNPs that are associated with lung disease, are they also associated with gene expression? And are they associated with gene expression for which of these genes under here, right? And here, what you can see is that these SNPs that are associated with lung function and this peak on the left are also associated with how much SLC9A3 expression one has, suggesting a putative mechanism here for why we saw association at this locus. But that's just a visualization tool. How do we test that? How do we test if indeed there is this co-localization that the same variants contribute? And this becomes a little bit challenging because you're in a region here where you have a lot of correlated SNPs. See all this color? You have two locations, two loci here, the blue peak and the colored peak. I told you we just did a GWAS where we had to do a meta-analysis. We had related individuals in there. We had lots of subtypes. And that's what those SNPs are associated, that's what those SNPs are calculated from, this complex model. So there are all kinds of ways out there to test whether the same SNPs that are associated with gene expression are also associated with GWAS. Tons of methods out there, but none of them can address the issue of when the SNPs are in high regions of linkage disequilibrium, and when you have multiple locations that are contributing at the same locus. You have overlapping samples, right? We're in a very complicated situation, and the tools out there cannot test for that. So we have Fan Wang, who is a doctoral student in statistics. She developed a test statistic that could do that. And this is really basically what we're asking, whether the pattern of SNPs that are EQTL, that are associated with lung disease, are also associated with gene expression. What is the evidence of this overlapping pattern? And so she developed a test statistic here where she uses a continuous measure of gene expression, and she integrates the gene expression with the GWAS association into this test statistic that she calls the simple sum. And she shows that we have type 1 error rate control. And she also shows that when we look at this test and we compare it to all the other tests that have type 1 error rate control, when you're in a simple scenario, like no overlapping samples, no meta-analysis, that it's the most powerful. And that's the line here with the red triangles. You can see that it's the most powerful test, Fan simple sum 2. So we have now a test that can accommodate all those complicated situations, that controls the type 1 error rate, and that is powerful. And so Fan worked with Naeem in the lab, who is a bioinformatician, to implement this in some software called Locus Focus. You can go ahead and try it out because you can integrate all kinds of different data. If you have a GWAS, if you have the p-values from a GWAS, you can take p-values from another GWAS, and you can upload them to Locus Focus and see if they're sharing genetic contributors to both of those phenotypes. And you can also do it with the gene expression. And so here's that visualization. And this is a heat map where it takes the co-localization test, and it tests across all the tissues and all the genes in the region. And it says, okay, is SLC9A3 co-localizing with the GWAS statistics across all of these tissues? And you can see here that indeed you see co-localization across many tissues for SLC9A3. And here is just when we implemented it, what our test statistic looks like. And indeed, SLC9A3 is associated with, and here's with the EQTLs, is co-localizing with EQTLs in the lung. And here's the heat map here, a close-up of that heat map, where you see all of the different tissues. 9A3 is co-localizing, especially here in the lung, which is the relevant tissue. You do see that it co-localizes with other genes as well. And what that means is that actually those variants that are EQTLs for SLC9A3 are also EQTLs for other genes, and that's quite common in the genome. So still lots of work to do at this particular locus. And so, so this co-localization approach allowed us to take the GWAS signal, right? And it allowed us to zoom into that GWAS signal and figure out what the putative responsible gene is, and the tissue, and it was in the relevant tissue of origin, and even what the possible mechanism was, which is improving gene expression. So with the last couple minutes, I'm just going to talk about extensions of the co-localization methods that we're working on. This is Eric Sanders. He's a PhD candidate in the lab. And he said to me, well, what if it co-localizes in males, but not in females? So what if there's a location, what if there's a genomic location and a phenotype that is more common in males than females? How does this co-localization stuff work? And well, everything would break down actually. So he started to work on ways that he could develop co-localization techniques for sex-dependent traits. And we can all think of many sex-dependent traits out there that would need specialized tools. And lastly, Henry Liu is a PhD candidate in the lab as well. We were starting to ask questions like, well, we saw that many tissues were co-localizing with SLC9A3. Well, maybe instead of testing one tissue at a time, we want to test groups of tissues that are across a common organ system or that are relevant. So he is interested in multi-phenotype co-localization methodology, and that's what he's working on there. So in conclusion, so co-localization here, we propose a summary statistics-based co-localization, visualization, and testing tool, and we implemented that in our software locus focus. You know, software implementation is data science as well, right? So that's why when we talk about genome data science, you know, and I mentioned all of the different aspects that go into genome data science, from data science projects in data preparation, in sampling and data collection, to creating tools and novel methods, right? And what's really helpful about the co-localization tools is that they allow us to go from GWAS and genomic locations to actual genes that we can put on that triangle plot and say, well, these are actual genes that are contributing, and this is how they're contributing. And now we even have drug development programs that we now, since we know the gene and we know that we want to augment the gene's activity, we can develop drugs that do that. So it really enables us to push that forward. So genome data science, you know, is a huge area. As technologies advance, we see that there are new challenges in genome data science. I told you that we have a huge amount of the program in the lab is taking new technologies and developing new informatics and new statistical tools so that we can, you know, we can really leverage these new technologies that come on the market. But this is not just in the genomic technologies. This is in storage, in computing, in the quality control, and then of course we do a lot of work in the data integration side. But you can't do anything and you can't solve any problems. I started with the talk today telling you that I wanted to solve a problem in cystic fibrosis, but we can't do that unless we have comprehensive, well-defined data, and we have engaged participants who understand why they're contributing to research, and they understand, and there's a constant communication between that population and the study teams. That is all a very important component of the data science effort. You know, I want to mention, of course, I think I've already driven this home, that GWAS just tells you about genomic locations. These locations could be very complicated. I started with SNPs, but I showed you we're missing so much variation in the genome. It's not just SNPs, it could be in very complex regions of the genome. So really, genome data science and GWAS are just the beginning. I hope I convinced you of that, of a long journey to understand the genetic contributions to the traits that we're studying. But it's not exclusive to genome data science. You could fill a genome with anything else, and you would see that the data science life cycle follows all kinds of application areas in a similar way here. So I will end so that we leave time for questions. I know we started a little bit late, but this is a humongous study. You know, I mentioned that we are enrolling individuals from all across Canada, and this has been going on for almost 20 years. But then, you know, we have collaborators from France and Italy and from the United States, so all over the world, really. And then we collaborate with all kinds of individuals who then take some of these modifier genes and they go and try to understand what the biology is behind them, as well as of course the genome sequencing. TCAG is our genome center where we collaborate with them to understand how do we generate this data. And then lastly, all the amazing students and staff that I've had the honor of working with, who've come up with some of these just absolutely amazing ideas that answer really important questions. And just that's such a privilege to work with them. So thanks, and I'm happy to take more questions.
So thank you, Lisa. That was great. And thank you for fitting it in the time. Sorry about the delay. Yeah, so we're happy to take questions. We do have a few minutes and we do have the room beyond this time too anyway. So I think I will kind of like pick out the questions from the audience in person. And Lynn, do you mind looking online? I know we already have a couple that have come in online. So Lynn will read those out. And yeah, if you're online, just feel free to add your questions to the chat.
Online first,
Let's do like one online first, and then we'll bounce.
Okay, so thank you for a great presentation. So the lab says, I'm wondering if you have any comments on using haplotypes rather than several million mostly imputed SNPs to improve statistical power and increase the likelihood of detecting causal relationships rather than mere correlation?
Yeah, so I mentioned that we were doing a lot of work in long read sequencing in sort of these new technologies, and the amazing thing about these new technologies is that you actually can observe phase. So you can observe those haplotypes that you're referring to. So you don't, because if you actually if you have SNPs, or you don't actually know, you have to estimate the haplotypes. But with these new technologies, what we can do is we observe them. And so we have started, I have a graduate student right now, he's doing amazing work where he's looking at developing statistical methods that account for and leverage phase so that you can indeed understand the complex relationships on a haplotype between variants. But of course, this is all still association. Just because you have haplotypes, you're still doing tests of association. And so in order to make causal interpretations, we still need to implement causal inference methods, or as well as trying things out in the lab, making changes and seeing what those changes cause. So even though we have phase, we are developing methods for analyzing haplotypes and testing phase relationships. They are still association.
Thanks for the presentation. My question's about the locus focus. First of all, fantastic name. Second of all, have you heard of any applications of it outside of genomics?
Yeah, so that's a great question. So I gave this talk at the University of Pennsylvania last year, and I had some neuroimagers in the audience, and they felt like that there were really good applications in neuroimaging. So it made me think that although I haven't used it outside of genomics, I've used it a lot of different types of genomic data and thinking about extending the sort of models across different types of questions. I think there's a lot of room there. I haven't applied it outside of the genomics area, but anytime where you have sort of a single unit that you're looking at the relationship between, so thinking about the brain, right? You have brain locations and you can associate them with different traits, then you can do that. You could apply locus focus and integrate from that. You need that sort of common SNP or location or whatever it is to be able to apply it, but otherwise it should work. Yeah, thanks.
So another question from Tony Desmond, one of our prof here at UC. What are the random effects in your mixed effects model?
Yeah, so our random effects, so for the GWAS, our random effects are the relationships and the platforms that we did the genetic association with. And then in some cases, it's also the genetic relationship matrix. So that is actually the approximation of the genetic ancestry.
Yeah.
Like, how do we know that it's better?
Yeah.
Yeah, so that's an excellent question. So we benchmarked it against CFTR mutations that we know. So everybody who has CF, right? Part of their diagnostic journey is to understand what their causal CFTR mutations are. And they do that in CLIA labs, and they look for all the known CF causing mutations. So we have that set. And so what we did is they're not in our data set, and we impute them, and then we compare them to what the truth is. And so that's how we're able to measure those, the accuracy that I showed you. Yeah, thanks. Mm-hmm. This one. Okay.
Do they have a timeline or they just random?
all of these different, the disease and the different, so that's an excellent question. So there are phenotypes that happen, at birth, actually. So there's an intestinal phenotype that happens at birth, and it's an intestinal obstruction. And we've done a lot of work trying to map the genes that contribute to that. Whereas there's CF-related diabetes, and that happens at an increased frequency as they age. So I think something like 33% of adults with CF have CF-related diabetes, but that's an age of onset trait. So there are different traits, different impact of age. The disease in the airways is a progressive disease that gets worse over time as they age. And one of the challenges actually is that children may have normal lung function, whereas adults, they start to see this decline in their lung function. And sometimes it's difficult to say, okay, well, I'm giving them a medication, did it work? Because they already started with very high lung function. So some problems like that. So there are age components to almost all of the phenotypes here. And so that is an important that's an important component, exactly. And it helps you to think about the timing of the data that you're looking at.
So in atrophy, so when some gene contribute to disease, right? So now you take it into some genes might involve in multiple gene expression, or some things involve in multiple gene expression. But you make concern about how the gene expression,
Yeah.
they have different expressions across different timelines, right?
Yeah. Yeah. So that's such a good question. I didn't get into this example. I skipped it because of time. But we have there's another location here on the X chromosome that I mentioned when we did this lung GWAS here. Okay, so we were focusing on this 9A3 locus on chromosome 5, but there's another locus here on chromosome X. And what was really interesting is that this locus here contributed to the lung phenotype, but also contributed to the intestinal phenotype in GWAS. There's no timing in GWAS, right? We just know, that the same location, general location, contributed to the both phenotypes, okay, in CF. So that's a bit, and what was puzzling here is if you can look at this plot, this is a bit difficult to digest, but these blue dots here are the zoom in location of association for lung disease. And these orange yellow dots are the zoom in association for the intestinal phenotype. And what you can see here are the two colored peaks for the two different phenotypes are actually at different locations here on chromosome X. So, most people would say, well, that's not a replication. It's not the same. It's different, right? Different variants are contributing to the different phenotypes. So actually, when we started to take gene expression from the pancreas, okay, and gene expression from the lung, and we did our locus focus plots, we saw that the lung disease GWAS hits were co-localizing with the gene expression of this gene 6A14 in the lungs. And the intestinal obstruction GWASs were co-localizing with gene expression of SLC 6A14 in the pancreas. So we had a common gene, but we have just a different regulation. And so this is pleiotropy. This classic, journals would give us a hard time about replication. Nobody would say that, there was actually debate in the field about whether the lung disease gene was AGTR2 and the intestinal gene was 6A14, because most people ascribe the closest gene as the responsible gene. So this is an example of how locus focus helps you to figure out that there's just a different regulatory mechanism here. And indeed, there's a lung specific enhancer that sits right here for 6A14. Yeah, it's nice. That was a nice story. Yeah.
So you kind of have like really similar gene expression that differentially expressed
like, are we looking at gene expression? So all the studies that I show you here, whether it's intestinal obstruction or the lung disease, are only done in cases, right? Because we're looking at comorbidity across the cases. But so the gene expression here, okay, in black in the pancreas and in brown here in the lung, are from healthy controls. That's from GTEC. So there's a lot of public data out there that sits in databases. One particular project that's extremely useful is the Genotype Tissue Expression Project. And what they did is they made gene expression for all genes across many different tissues of the body from controls available in repositories, and we have all the EQTLs for that. So here we can use that data to show that this locus is a pancreas locus. But then the blue line here is the gene expression from the CF population. So you can see that the blue line and the brown line agree, but not as it's much stronger in the CF population. So yeah, no, it's definitely important to be able to bring together the public data as well. It also tells you about, in the absence of that causal gene, what's the impact. Yes.
So I'm assuming that you have CF data and healthy data also, and CF data will be very less compared to the healthy data. So is there any imbalance in the data, or you have problem there? Like ones are very less and healthy are.
Yeah, so in this particular study, we're only using the CF because we're interested in, are there genes that make this CF individual's lung disease worse than that CF individual's lung disease, right? Or that makes this CF individual have an obstruction in the intestine and not that. So it's all analysis within the CF population. But we have a lot of other studies in other diseases where we use public controls, exactly as you said. And if you're looking at a case control analysis with public controls, and there are hundreds of thousands of controls and a few thousand cases, you definitely have to think about case control imbalance. And there's all kinds of study design questions. And that goes into all that data science thinking as you prepare your data and you design your study. So those are all really important questions. Yeah. Yes. So just to repeat the question, and for the online audience as well, as if I got it right. So are you asking whether we have information not only about their CF disease, but about other aspects of their health? Like, do they have other chronic conditions and that sort of thing? Yeah, so we have in the registry, there's all kinds of information about their general health as well. So we do know that, but this is a very different population. Like CFTR is expressed in many different cell types across many different organs. So it's difficult sometimes to figure out whether the chronic disease that you see, right, is CF specific, or is it specific in the general population? So for example, we have a study right now with a collaborator at St. Mike's Hospital interested in bone disease in CF. But the assumption is that CFTR plays a role in that as well. So there's this constant disentangling of when you're studying some of these chronic diseases that you see in the general population as well, how much of their susceptibility is contributed to by genes that contribute to that disorder in the general population versus CF specific. And we've written a few papers about this, and that's actually one of the reasons I mentioned that one of the goals of our study is also to understand how the modifiers contribute to disease in the general population or traits in the general population. Thank you.
Okay, so I think that's all we have time for today. Thank you so much for all of the great questions, though, and thank you again, Lisa.
Pleasure.
So just a couple reminders that today at 2, all the students, there is a drop-in session with Lisa, so if you do have more questions that you aren't able to ask, feel free to drop in and you can ask her anything you would like. And then for faculty, the session is at 3, and both of those are in this room. So we hope to see you all there as well. And thank you again, Lisa.